Hello everyone! This video covers Chapter 7 in our textbook, Essential Biochemistry, Sections 7.1 and 7.2. So before we get started, I wanted to give you an overview of rate laws, and this is a review from General Chemistry. You might remember from General Chemistry that rate laws are written in terms of reactants and they're determined experimentally, not stoichiometrically. So that means that somebody can't give you an equation and just say, tell me the rate law. They would have to give you experimental data. So I got these reactions from a General Chemistry book. They're obviously gas phase reactions. So here we have the decomposition of N2O5 to NO2 and an oxygen atom. And the rate law was determined to be rate equals K times the molar concentration of N2O5. So you can imagine this is raised to the first power. So that means that the reaction is considered to be first order in N2O5, first order overall, and we can see that the rate is proportional to N2O5 because if you double the N2O5 concentration, you double the rate. If you triple the concentration, you triple the rate. The other thing that this tells us is that you get a rate law like this because the rate limiting step is unimolecular. What that means, this is the mechanism right here, is that if we look at this reaction, the mechanism, which happens to be a three-step reaction, this is how it takes place. The N2O5 collides with some molecule M and forms a higher energy state. And then that decomposes to form NO2 and NO3. And this is the slow or the rate determining step. And it just relies on N2O5. So that's why the rate law tells us that the rate is dependent upon the concentration of N2O5. The next step is rapid and it involves the decomposition of NO3 to NO2 in the atom of oxygen. So that's our slow step, and the rate limiting step, the rate law is always based on the slowest step because the rate of the slowest step will determine the rate of the reaction overall. So if I went into the lab and I did this reaction, I could take the concentration of the N2O5, I could measure the disappearance of that reactant over time. So the velocity or the rate of the reaction could be expressed as the disappearance of the reactant over time, and there's my rate law right there. If I integrate the rate law, I get this equation right here, which is in the form of a straight line. Y equals mx plus b. So what that means is that if I plot my data, if I plot the natural log of the concentration of my reactant, in my case N2O5, and it disappears over time, I could measure the slope, and the slope is equal to minus K. So my K, I would just take the opposite sign because K's are always positive. So I could obtain my value of K from the slope. So let's look at another example. This is the decomposition of ethane, and we would go into the lab and we would do some experiments and find that the rate is equal to K times C2H6 squared. So this is a second order reaction. The reaction is second order overall, and the rate is proportional to C2H6 squared. So if I double the concentration, I quadruple the rate. If I triple the concentration, I increase the rate by a factor of 9. I don't have the mechanism of this reaction, but I do know from looking at the rate that the reaction rate, that the slowest step must somehow involve the collision of two molecules of C2H6. That's what the rate law tells me because it's a bimolecular reaction. So here, if I were to measure the disappearance of ethane over time, and this is the rate law that I would get, again, experimentally, and if I integrate it, I again get y equals mx plus b. So this is a different form. This is the form of a second order rate equation. So if I plotted the reciprocal of my reactant over time, I would see that I would get a straight line, and here my slope is equal to K. So that's how I would solve for K in this case. I can give you this example here. We can have a second order overall reaction where you have two different reactants. So here I have the reaction of NO plus O3 gives NO2 plus O2. So again, it's first order in NO and first order in O3, so the rate is proportional to both NO and O3. It's second order overall, and again, I don't have the mechanism of this reaction, but I know that it has to be bimolecular, and I know that it involves the collision of an NO and an and O3. And that's the slow step, because the slow step will determine the rate of the overall reaction. I can also give you a rate law like this, where I have rate equals K times NO squared times O2. And here we see that it's second order in NO, so the rate is proportional to NO concentration squared. It's first order overall. Sorry, it's first order in O2, but it's third order overall. So you might conclude that you have a termolecular reaction. Well, this rate law is consistent with that, but it's not likely. It's hard to imagine two NOs and one O2 colliding with sufficient energy and the correct orientation in order to accomplish a reaction. So this is what you get mathematically if you solve for the rate law when you have a situation where the first step is rapid and the second step involving an intermediate is slow. And we have to write the rate law based on the slow step, but also remember that rate laws are not based on intermediates, they're based on reactants. So that means you have to algebraically solve for an expression that gives you the rate law in terms of the reactants. So if you see something like this, it's not termolecular. This is the likely scenario. So in fact, reactions are either unimolecular or bimolecular. Termolecular reactions are really not heard of. So let's go to another example right here. You might have two reactants in your reaction, but the rate law is based on only one of them. So here we have an example involving T-butyl bromide, where the reaction is first order in T-butyl bromide and first order overall. But it's zero order in hydroxide. Now, hydroxide is obviously important to the reaction, but what this means is that the rate limiting, or the slow step, is unimolecular. It only involves the T-butyl bromide, and it doesn't involve the hydroxide. So the hydroxide must be involved in a fast step, which is why it doesn't appear in the rate law. So let's get to some biochemistry. Suppose I give you a reaction where sucrose is hydrolyzed. So the rate law here experimentally shows us that the reaction is dependent on the concentration of sucrose and the concentration of water. It's first order in both of these and second order overall. The slow step is bimolecular, involving a collision between the sucrose and the water. But in actuality, the reaction really is not really dependent on water. And that's because the water is a reactant, but it's also a solvent. So in reality, the reaction rate is really only dependent on the concentration of the sucrose that you have, because you have so much more water. So we say that the reaction behaves as though it were zero order in water, because the concentration of the water doesn't affect the rate because the concentration is so high. So we would call this a pseudo first order reaction, because it really is a second order reaction, but we could say it's pseudo first order. Now, if we were to do this reaction and throw an enzyme in there, we'd have to completely write the rate law, because when we have an enzyme, the mechanism is altered in the presence of the enzyme. So that means that the rate law has to be altered as well. And what happens with an enzyme catalyzed reaction is the enzyme first has to bind the reactants, and we refer to these reactants as substrates. So let's segue from there into general properties of enzymes. Enzymes are catalysts. Their reaction rates are higher, but it's important to note that the free energy values and the equilibrium constants for the reactions are the same with and without the catalyst. You just get there faster. We accomplish the reaction in milder reaction conditions. It's not like being in an organic chemistry lab where you can add a strong acid or a strong base as a catalyst. We have to do this at pH 7.4. Also in the lab, you might carry out a reaction and get a number of different products and spend a lot of time purifying the reaction mixture to get the product you want. We don't do that in the cell either, generally. Generally, we get only one product. And nearly all enzymes are globular proteins. So at one point I would have said all enzymes are globular proteins, but in the late 80s it was discovered that there are some ribozymes, RNA, that has catalytic activity. In this class, though, we will focus on proteins as enzymes. So we are going to start out with a Fisher's lock and key hypothesis, which is not strictly accurate, but in terms of what we're doing today, it's a fine model for us. We can envision that the enzyme, so here we have a folded protein where we have an active site, and we have a substrate, a reactant that binds to the active site on the enzyme. So we consider the enzyme to be the lock and the substrate to be the key. So the substrate binds to the enzyme's active site. And we form this enzyme-substrate complex. So this is really sort of a bimolecular reaction. I have reaction in quotes. It's not really a reaction because when the enzyme binds to the substrate, it does so via non-covalent interactions. So all of those IMFs we've studied, London dispersion forces, electrostatic interactions, hydrogen bonding, hydrophobic interactions, all of those are important when an enzyme binds to the substrate to form the enzyme-substrate complex. We can illustrate that with this figure right here, where we can show that when a substrate binds to an enzyme, we have both geometric and electronic complementarity. So geometric means that the substrate's shape will fit into the shape of the active site. Normally there's sort of a cleft on the surface of the enzyme that's whose shape is complementary in shape to the substrate. But we have electronic complementarity as well. So if we have our substrate right here, this little brown H is to represent hydrophobic interactions. So we have hydrophobic interactions between our enzyme and our substrate. These blue dashes right here are hydrogen bonds. So maybe we have a hydrogen bond donor on our substrate and a hydrogen bond acceptor on our enzyme, or maybe it's the other way around where we have a hydrogen bond donor on our enzyme and a hydrogen bond acceptor on our substrate. Or we could have ion pairing where we have a negatively charged substrate and a positive charge on our enzyme. So all of that would be considered to be electronic complementarity. So let's start with a simple enzymatic reaction in which we consider only one substrate and one product. So we'll have our enzyme binding to our substrate to form the enzyme-substrate complex. We'll use the rate constant K1 to describe how the enzyme and the substrate bind to form the enzyme-substrate complex. This is a reversible process, so we'll use K to the -1 if the substrate dissociates from the enzyme. And again, non-covalent interactions are what are involved here. We'll use K2 to describe what happens when the substrate is converted to product. The substrate conversion to product will involve breaking bonds in the substrate and forming new ones in the product. So we'll have covalent bonds being broken and formed here. And that process we'll use K2 to describe that. And we regenerate the enzyme at the end like all good catalysts do. So I'm going to describe the work of Michaelis and Menten. So this is Michaelis and this is Menten, and they worked together on enzyme catalyzed reactions. And this was back before they even really knew anything about enzyme structure. So what they did is they chose an enzyme catalyzed reaction in which the product had a color to it. So you can measure the reaction velocity by seeing how fast the colored product forms. So in this reaction right here, I have an inset where we've got 1, 2, 3, 4, 5, 6, 7, 8. So you'd run 8 different reactions. So every point is a different test tube where you'd have an increasing amount of substrate, and you'd keep the enzyme concentration constant. And they found when they plotted reaction velocity versus substrate concentration that they got this nice hyperbolic curve. And you might remember from your math class that y equals ax over b plus x is the equation of a hyperbola. So that's what you get when you go into the lab, is you get a hyperbolic curve. So what we should do now is get out our pencils and paper and derive an equation based on our theoretical understanding of how enzyme binds to the substrate. So we can do that by saying that the velocity of our reaction is dependent on this rate constant K2 and the concentration of the enzyme substrate complex. Now we're not done, because remember when we started this lecture we said you can't write a rate law based on an intermediate. The enzyme substrate complex is an intermediate. We're going to have to do some algebraic manipulations to describe our rate law in terms of enzyme and substrate, which are our reactants. So rate laws have to be written in terms of reactants, so we can't stop there. Okay, so what we're going to do first is we are going to assume steady state when we're looking at our enzyme substrate complex. So, and that's going to be true pretty much all the way through the enzyme catalyzed reaction except for the very beginning and the very end. So the rate of formation of ES is going to depend on how much enzyme and substrate you have, and this rate constant K1. And it's also going to depend on the rate of breakdown. When ES breaks down, it can go in two different ways. You can just have the enzymes dissociate from the substrate, or you could have the substrate be converted to product. So both of these constants are important, and they will depend on the concentration of the enzyme substrate complex. So we're going to assume steady state, in which we're going to say that the rate of formation of ES equals the rate of breakdown of ES. So we do that. So if this equation represents the rate of formation of the enzyme substrate complex, and if this equation represents its breakdown, then we're going to set them equal to each other. Okay, so here's that equation that I just showed you. So mathematically, I'm just going to solve for ES. So I get this expression right here. And now I'm going to define Km. So we're just going to collect all of our rate constants and have one big constant called the Michaelis constant. And that is equal to K minus 1 plus K2 over K1. So if I've defined Km, then I can substitute K minus 1 plus K2 over Km, and that puts, K1, sorry, and that puts Km in my denominator. So I have ES equals the concentration of E times the concentration of S divided by Km. So the next thing I need to do is I need to express my terms and things in terms of things that I can measure. If I'm setting up my experiment, I know how much total enzyme I put in my test tube. But once I put the enzyme in the test tube and the substrate is there, I don't know how much of the enzyme binds to the substrate and how much of the enzyme is free and is not bound to substrate. But I do know that the total enzyme concentration has to be the amount of enzyme that's bound to the substrate plus the amount of the enzyme that's not. I do know that. So let's then solve for E. So this is just enzyme that's not bound to the substrate. That's equal to the total amount of enzyme subtracted from that, the amount of the enzyme that is bound to the substrate. So remember on my previous slide, I had this expression for ES, and I did have this E term in here. And again, that E is enzyme that's not bound to substrate. And again, I don't know what that is. So what I can do is I can just put this expression right here. So ET minus ES, I can put that in my expression right here, and that way I get rid of a variable. So now I have ES expressed in terms of the total amount of enzyme and the substrate. I'm going to do some more algebraic manipulations because I want to find an expression for ES, and I've got ES on both sides of the equation. So I'm going to solve for ES, so I'm not going to show all the steps involved, so that's something you can do on your own. But what I have now is an expression for the enzyme substrate complex, an intermediate, a quantity that I can't measure, in terms of quantities that I can. I know how much substrate I added to my test tube, and I know the total amount of enzyme that I added. So now, if I go back to this rate law that I wrote right here, where the velocity or the rate of my reaction is dependent on the concentration of the enzyme substrate complex times K2, I have now an expression for ES that's written in terms of reactants. So now I can write my rate law because remember the rate law has to be written in terms of reactants, not intermediates. So there is my rate law that I wrote. There is the expression for ES. So I can take all of that and substitute in, and now I have an equation that describes my enzyme catalyzed reaction at all points along the curve. So I want to convince you that that's true. So what I want to do is suppose, let's look at our curve where the velocity is equal to the maximal velocity. So at the maximal velocity, the substrate concentration is far greater than the Km. So if that is true, then in my denominator, if this Km term is very small compared to my S term, I can write an approximation in which I cross out my substrate concentration, and now I have Vmax equals K2 times ET. So that makes sense. So the fastest that reaction can go is going to depend on how much enzyme I have and how fast the substrate's converted to products, and that's described by K2. So what I can do then is I can take this K2 times ET and I can substitute the term Vmax in there. So now I have the Michaelis-Menten equation, and it's V equals Vmax times S over Km plus S. Well, you might notice that that's an equation of a hyperbola. That's Y equals AX over B plus X, and that's an equation of a hyperbola. So what that means is that if I go into the lab and I do an enzyme catalyzed reaction and I get a hyperbola, and then I get a pencil and paper and derive an equation that's based on my understanding of how enzyme catalyzed reactions occur, and if they match up, that means my understanding must be correct. So I get this hyperbola right here. So my simple reaction, which I have one substrate and one product, involves the enzyme binding to the substrate to form the enzyme-substrate complex. The enzyme is regenerated, the substrate is converted to product. And I get this hyperbola right here. And now I can define these terms in terms of the reaction this described by this hyperbolic curve, that when I get to high substrate concentration, I'm going to approach Vmax, the maximal velocity, as an asymptote. The other thing I can do is I can define Km, so that's the B term in my Y equals AX over B plus X. So if I go to 1/2 Vmax, draw a line down, draw a line over, and draw a line down, I'll get the value of Km. So if we look at this, I do want to convince you that this equation describes all of the points along the curve. So if we look here at low substrate concentration, what we find is that the reaction is first order in substrate concentration, that this is approximately linear, which means as I increase the substrate concentration, I get a proportional increase in velocity. And that occurs, again, based on my understanding, because if the substrate concentration is low, I have a lot of enzyme, and that means that all the substrate is being converted to product, and the amount of enzyme isn't limiting me to how that happens. So that means if I increase the substrate concentration, I can increase the reaction rate. And again, we're assuming that all of these experiments right here, represented by each one of these points, the enzyme concentration is constant. So we would say that the reaction is first order in S, and we would say that it's zero order in enzyme, because at this point the enzyme is present in a high concentration, so that means that it doesn't really matter how much enzyme that I have. And again, the other thing that we should note is that if we're looking at 1/2 the maximal velocity, that we get our definition of Km. So Km is that substrate concentration at which the velocity is equal to 1/2 the maximal velocity. So look at the asymptote of your curve, look at the halfway point, draw a line over, drop a line down, and that's the Km. So Km always has units of substrate concentration. So let's look out here now. What if we're way up here at really high substrate concentration? So again, if we're looking at our lock and key model, here the enzyme is completely saturated with substrate, and you've got substrate left over because the substrate concentration is so high and it's greater than the concentration of the enzyme. That means that increasing the substrate concentration doesn't affect the reaction rate. So you're approaching the Vmax because all of the substrate is bound to all of the enzyme, and the enzyme is working as hard as it can to convert substrate to product, and having extra substrate isn't going to make the reaction occur any faster. So we approach the maximal velocity. So here what we get is a situation where the reaction is zero order in S and first order in enzyme, because increasing the substrate concentration doesn't impact the rate. So the reaction is really zero order in substrate concentration when we get to the really high substrate concentration. So that's our Vmax as our asymptote, and the Km is the the substrate concentration at 1/2 Vmax. So remember earlier we defined Km as K to the minus 1 plus K2 over K1, and we got these Ks right here from our simple understanding of how enzyme binds to substrate, forms the enzyme substrate complex, converts substrate to product, and regenerates the enzyme. What we should note is that generally K1 and K minus 1 are larger numbers because it involves just the association of the enzyme and the substrate to form the enzyme substrate complex, or the dissociation. And those are non-covalent interactions, where the K2 is going to be a lot slower because you're breaking covalent bonds in the substrate and forming new ones in the product. So what we could do is we could have sort of an approximation where we approximate Km as being a ratio of K minus 1 over K1. So what that means is that in simple cases, we should emphasize this, in simple cases, Km is a measure of binding affinity. Because we can see if we have a high K to the minus 1, if our dissociation constant is high relative to the K1, then the affinity of the enzyme for the substrate is going to be low. But if our K1 is high compared to the K minus 1, that means that there's a tight association between the enzyme and the substrate, and the affinity of the enzyme for the substrate is going to be high. So again, this is an approximation, and this occurs for only the simplest cases of enzyme binding to substrate. So again, let's just look at our simple system. So here's our K2. So K2 is the rate constant of the conversion of the enzyme plus product from the enzyme substrate complex. So for simple systems, K2 is equal to Kcat. So we can solve for Kcat. So Kcat is Vmax over ET, and if we look for how we might put our values in here, you could express Vmax in terms of, say, molarity per second, and the enzyme concentration in molarity, and what that means, what you get for Kcat is a number divided by second. So that's sometimes known as the turnover number. So if you had, say, 100 per second, that would just tell you how many cycles of that the enzyme is converting substrate to product in 1 second. So that's sometimes known as the turnover number. In published papers, you'll often find Kcat over Km, and this is called catalytic efficiency. So often you'll see these values reported. So catalytic efficiency is Kcat divided by Km. So again, if we look at the units, if Kcat is reciprocal seconds and Km is in our denominator, then again we just have a number divided by molarity, and seconds are the units in the denominator. So this describes how often the enzyme and the substrate encounter each other in solution, because that's described by Km, how avidly they bind, and how rapidly substrate is converted to product, because that's described by Kcat. Now, there's only so high that you can go because the enzyme and the substrate can't encounter each other faster than diffusion will allow them. So enzymes approach catalytic perfection if the Kcat over Km approaches the rate of diffusion, which is about 10 to the 9th. So if you see values up right here to 10 to the 7th to 10 to the 8th, you get enzymes that are approaching catalytic perfection, and that they're working as fast as they possibly can do so, based on how fast the enzyme and the substrate encounter each other in solution. So how do you measure Vmax and Km? It's actually a lot easier now than it used to be years ago, because curve fitting programs to fit data to hyperbola are widely available and inexpensive or free. So you can take your substrate concentration and velocity and then fit your data to a curve, and then the program will usually give you the value of the asymptote, and then the 1/2 Vmax dropping line down is the Km. So that's easy to do. Now, it didn't used to be easy to do. So what was done before those curve fitting programs were either readily available or low cost or free, was that people would use Lineweaver-Burk plots. So you still see them in published papers, which is why I'm going to tell you about them. So here, we take the reciprocal of the Michaelis-Menten equation, and we get the equation of a straight line. So we get y equals mx plus b. So what that means is that instead of plotting velocity on the y-axis and substrate concentration on the x-axis, we're going to plot the reciprocal of the velocity versus the reciprocal of the substrate concentration. If you do that, then your y-intercept is 1 over the Vmax, and the slope is Km over Vmax. Now, if you solve for the x-intercept by taking y equals mx plus b and setting y equal to 0, you'll find that the x-intercept is minus 1 over Km. So a Lineweaver-Burk plot looks like this. So what you need to do is you have all of your substrate values here because you can't have negative substrate. So you'll get a straight line, and the line will cross right here at 1 over v. So the y-intercept is 1 over Vmax. So what you do is you look at your y-intercept, take the intercept, and that gives you Vmax. The slope is equal to Km over Vmax. So you could then substitute in the value for Vmax and then solve for Km. But an easy way, I think, is to solve for the x-intercept. And notice that if you extrapolate back to the x-axis, that the x-intercept is equal to minus 1 over Km. And that's how you can solve for Km. One thing that we should point out, which is why a lot of my colleagues prefer to use the curve fitting to the hyperbolic curve, is that you do have a lot of error here in the Lineweaver-Burk plot, and the error is especially high when you get to high values of 1 over the substrate concentration, which is low values of substrate. So this is just something to keep in mind when you look at Lineweaver-Burk plots in published papers. But if you're given a Lineweaver-Burk plot, it is, I chose a simple example right here. So if I plot my data and extrapolate back, and what you would have done is, say you've measured your velocity in micromolar per second, what you have here is you have seconds per micromolar, and that's indicated by this minus 1 right here, because if you take the reciprocal of the number, you have to take the reciprocal of the units. So if we cross over here at 0.02, then if we take the reciprocal, we get a Vmax of 50 micromolar per second. We can do the same thing over here for the Km. We can see that our x-intercept is equal to minus 0.04. And again, if we take the negative reciprocal of minus 0.04, we get 25 micromolar. So again, on our x-axis, we would have plotted 1 over micromolar, the reciprocal of the substrate concentration. So that's the basis of a simple enzyme catalyzed reaction. So thank you for your attention, and I will see you in class.